That's a great question. And so for why I think 206 kind of we're at the right time is really related to some of the things that we're able to overcome, uh, particularly as I kind of mentioned earlier, the design-based approach. And so in the kind of anti-infective or antibacterial space, peptides have had a challenge mostly associated with toxin safety. So when you put them into humans, they're not terribly safe. And that was one of the things we tried to specifically address with 206 during the design process was how could we design the molecule to essentially decouple the activity characteristics, so the good attributes of it from the toxin safety problems. And, all of the data that we have till now has showed that we have been able to do that. So we don't see any of the toxin safety concerns. So I think really the main driver of kind of why 206 is good now is because we've really been able to demonstrate clinically, and so in humans, that we don't really have a lot of the same toxin safety concerns that have plagued this class of molecules for since their kind of early discovery in the 1980s. The biggest kind of milestones that are upcoming is really in a couple things. One, it's always exciting to get the drug and treat your first patient, which we're planning for and shooting for later this year. And then I think the next big milestone would be the phase two data. So we're doing this as a phase two, three adaptive study. And so getting that phase two data, which will allow us to then do a sample size re-estimation for the phase three, that's the next real big milestone.